Grazie tanto per questa introduzione, per questo invito. Mi scuso se non parlo in italiano, se parlo in inglese, perché dopo tanti anni che manco qui, e specialmente dopo un viaggio così lungo, l'italiano è un po' arrugginito, quindi sono sicuro che non avrete nessuna difficoltà a seguire questo discorso. So I guess we can get started. I don't think it's really so critical at this stage. So I'm sure that everybody is familiar with the concept of angiogenesis, which is growth of blood vessels. It's obviously an extremely fundamental biological process. I guess it's very difficult, to imagine that you have any growth, process beyond an extremely small size, unless you have, growth of blood vessels. So it's primarily a physiological process, very critical for embryogenesis. But in all fairness, most of the interest in angiogenesis has been historically, traditionally centered around some pathological condition like a tumor growth. Actually, this is one of the very early papers. If you're interested in the history of angiogenesis, this is a very seminal paper, a very fundamental paper published actually in 1939. So some early investigator actually were using what at that time was a very state-of-art technology, which is a transparent chamber. So you can put your tumor or whatever tissue you want, in a glass chamber. Oh, perfetto. Thank you. Perfetto. Questo qui. Sì. So in this transparent chamber, in this case, they introduce a tumor, and actually they observe, as you can easily see, this tumor is in the middle, and then you have this growth of vessels, this tortuous vessels, serpiginous vessels, which grow radially toward, the tumor. And this investigator actually very insightful, hypothesized that the tumor must be elaborating some substance, some blood vessel growth, stimulate. And so this was the idea that there is some biochemical mediator. Actually, at that time they were called, chemical mediator. Was an idea which was already established in the beginning of the century. Probably the first, paper which clearly makes this point was published in 1913. So the idea that, there is some biochemical mediator underlying some important biological process was not really new. But certainly this investigator was very insightful, in hypothesizing that the tumor produces some molecule which induce, blood vessel growth. And of course, there was some, in the period it had been extremely difficult to do any follow-up in the 30s because clearly the biochemical or let alone the molecular tools did not really exist at all to do that, And actually, we need to go to have some progress in this field, to go back to the 70s and to the 80s, when actually it became clear there is many angiogenic factors, many factors which can produce angiogenesis, at least when they're testing some system, like, in bioassay. So at least as a pharmacological agents, there was already many factors which can induce angiogenesis. Probably the best known at the time was basic FGF, which was a very potent molecule which can induce angiogenesis in a very effective way, and a series of other molecules. But in this review published in 1991, which is very, very insightful, it was already clear that even though this molecule can potently induce angiogenesis in a number of assays, it's not clear at all whether they play a role as an endogenous mediator. Just to make you an example, in the late 80s, people develop antibodies to FGF, with this extremely potent molecule. To everybody's surprise, this antibody did nothing in terms of inhibiting tumor growth. And actually later on, the knockout of basic FGF, even the double knockout acidic and basic FGF, did not have any phenotype. So very clearly something was missing from this picture, some molecule which remained to be discovered. That's why I was very fortunate, actually to have an opportunity to give a scientific contribution in this area. Actually, I started working, in a very different field. I was working in endocrinology. I was a postdoc at UCSF in the Reproductive Endocrinology Center, actually. And was as a part of my postdoctoral project, I was studying some really poorly known cells from the pituitary gland called follicular stellate cells, which at the time were not even of a very uncertain function. But the cells, actually, I wish I had a slide to show them. I forgot. The cells have a very unique organization in the pituitary. They resemble to astrocytes. They have lots of projection, which and very closely to the perivascular space, to the vascular basement membrane. And so this, even some early morphologists speculated these cells play some role in the maintenance of the pituitary vasculature. And actually, at the time, I was able to test, the supernatant from follicular cells into endothelial cells. And I found that at the time, to my surprise, they were strongly induced endothelial cell growth, And this, was, of course, was an observation. This happened at the time when basic FGF was discovered, and at the time everybody thought that this activity is very likely due to basic FGF. My state that was not true. I was able, to isolate, this molecule, purify at the time by conventional protein biochemistry, and actually this was I did this work when I was at UCSF, and then I joined Genentech, which at this really incredible biotechnology company. At the time, it was really at the cutting edge of the technology. And in a very short time, in fact, we were able to determine not only an N-terminal amino acid sequence of this protein, but based on the amino acid sequence, actually, we designed some probe, to clone the cDNA. This, of course, was a technology of 20 years ago. Now all the genes are known. There is very little, nothing to discover in terms of factor. But in those days, there was still something really unknown that was interesting to discover. And so this is actually, I'm not going into any details, which probably you may not find to be particularly interesting. The point is that, we cloned the human and bovine, cDNA, actually. And there was a very high level of homology. There is about 95% identity between these two species, which certainly suggests that this is a conserved molecule. And actually, this is all. This is summarized some of the characteristics of. Actually, we call this molecule vascular endothelial growth factor, because unlike, for example, basic FGF, or the molecule I showed you previously, which there seems to be broad spectrum mitogens. For example, FGF, as the name implies, is a mitogen for fibroblast. This molecule actually, at the time, seems to be very selective for endothelial cells, which of course is the key cell type in the vasculature. Among a very panel of cell type, they seem to have mitogenic activity, at least selective for endothelial cells. So that's why this name, vascular endothelial growth factor. And I think the biologists proved that this was a correct denomination. And this molecule has a number of characteristics, like it's a molecular weight of 45 kilodalton, it's a glycoprotein, etc., etc. And so this is another really point which probably is not worth going too much detail, because unless you're an aficionado in this field, this is the human VEGF gene. This is the gene. There are 8 exons which happens. It's very common. Genes have multiple exons, and there is by alternate exon splice you can generate multiple isoforms, multiple molecular species. Actually, there are as many as 7 or 8. This is the original isoform of VEGF. You can see there is they encode for protein with rather a spectrum of size, from 121 all the way to 106 amino acids. But the key point is in the size is the difference in affinity for heparin. VEGF 121 lacks affinity for heparin. Instead, VEGF 165 is one heparin binding domain, which is encoded by exon 7. And then VEGF 189 and 206 have each 2 heparin binding domains. So this creates like a potential a spectrum of polypeptides which have a differential affinity for heparan sulfate. And so essentially, which we're able to show that this result in molecules which have a spectrum of diffusibility, bioavailability. VEGF 121, which does not really bind to this component, is the most diffusible molecule. VEGF 206 is so tightly bound to the extracellular matrix that essentially is practically insoluble, is not diffusible. And then the most important component is VEGF 165, which is partially bound to the extracellular matrix and partially diffusible. And so now it's very clear that this diffusibility, this differential diffusibility, is critical to develop some tridimensional gradients, of angiogenesis, because angiogenesis is a complex in vivo phenomenon which rely on attracting vessels. And so this spectrum of polypeptides seems to be very well suited to do so. And there is another mechanism of regulation of bioavailability of VEGF, which is in parallel to the alternative splicing, which is extracellular proteolysis. Actually, there is now, it's very clear, there is a series of proteases, which are physiologically relevant to proteases, which can cleave VEGF. Actually, this is one which is plasmin. A member of the MMP, matrix metalloproteinases, can do something very similar. Actually, plasmin cleaving VEGF, in the carboxyl terminus, and generated first, this heterodimer, and that is homodimer, VEGF 110, 110. So essentially, what this does is convert, a heparin binding polypeptide into a shorter fragment which is very diffusible and essentially is essentially biochemical and biological. It's very similar to VEGF 121. And actually there is evidence that this phenomenon is especially important from a pathophysiological point of view. For example, in situations like age-related macular degeneration or even in tumors, this may be the final mediator of the activity of VEGF, especially when there is actually activation of proteases. Actually, now we understand a lot more about VEGF than we did in 1989, etc. Now we know that there is a series of receptors. There are two main tyrosine kinase receptors for VEGF. Actually, as you can see, there is now lots of molecules. The original VEGF now is referred to as VEGF-A and interacting with the VEGF receptor 1 and VEGF receptor 2. And also, other investigators discovered other molecules like PLGF and VEGF-B, which interact selectively with the VEGF receptor 1. Actually, PLGF was discovered by an Italian scientist, by Graziella Persico, who unfortunately passed away a few years ago, but she was a very, very, very good scientist. And then VEGF-C and D, we actually primarily interact with the VEGF receptor 3. In interest of simplicity, I'm not going to talk about the system, but VEGF receptor 3 is very important in that regulating another aspect of angiogenesis, which is lymphatic angiogenesis, lymphangiogenisis. Instead of the VEGF-A, it primarily regulates hemangiogenesis, the classical angiogenesis. And now it's very clear actually the VEGF receptor 2 is the main receptor which mediates most of the activity of VEGF, which I was illustrating at the beginning, like mitogenesis, angiogenesis, and permeability. VEGF receptor 1 is still, it's a very complex molecule which can do really very different things depending on the context. Actually in early development it's very paradoxical, it's an inhibitory system because it can bind VEGF with such a high level of affinity, essentially prevent from interacting with the VEGF receptor 2. But if anybody is interested I'll be happy answering the question. But the point that the most important point is VEGF receptor 2 is the main molecule, is the main therapeutic target, as I will show you. Of course, when you have all this molecule, actually, the first thing that comes to everybody's mind, is that there is a very complex, system where there is a lots of redundancy. There is many components. So essentially, in theory, it would be very difficult to identify the key molecule. You could, easily postulate that if you, let's say, block, VEGF-A, VEGF-B or VEGF-C can replace, the function of VEGF. And that's what many people believe. But this experiment essentially completely contradicts, this notion, actually. This is the knockout, of VEGF-A in mice. Of course, you're very familiar with this technology. And essentially, what is this is the yolk sac, mouse embryo, day 10.5. This is the wild type, which is both copies of the VEGF gene. You can see this very nice vascular pattern, and this is the embryo which is underneath. This is the mutant which lacks only one of the VEGF alleles. You can already see that you can hardly see any blood vessels, any blood islands, and then at the stage it's already underdeveloped. It's probably already dead. So the point is that even loss of a single allele of VEGF is incompatible with embryonic development, which is it's an extremely rare phenomenon. There is probably one or two other examples in the mammalian genome or genes which seems to be so critical, even haploinsufficiency result in embryonic lethality, which is also equally interesting if some investigators have done some genomic manipulation in such that you have a slight increase in the level of VEGF, 50%, and that too is incompatible with embryonic development. So at least during embryogenesis there is such a tight regulation that probably because you need to create this correct tridimensional gradient. Such if you deviate in excess or in defect, that is really detrimental. So once again, this is another concept of vascular biology which has emerged over the last several years. The fact the VEGF is very tightly regulated by another very important system, which is Notch. Notch is a very fundamental family of regulators in embryogenesis, another circumstance. In particular, delta 4 Notch, DLL4 Notch, essentially inhibits VEGF. It's like a brake for VEGF, like negative feedback, such that if you block Notch, there is this excessive activity of VEGF, resulting in this filopodia, which are called now tepinocells, And you have this, if you block Notch, you have this excessive sprouting, which results in non-productive angiogenesis. Actually, this approach has been tested even to block tumor growth, because it results in this non-productive angiogenesis. But once again, a very key concept is to appreciate there is this kind of functional relation between Notch and VEGF. And so, of course, there's been since the very early study, there's many studies which have shown the importance of VEGF in many physiological processes, which I'm not going to show you in the interest of time. For example, not only in embryonic development, even in the adult, like in reproductive angiogenesis, like in corpus luteum angiogenesis, in skeletal, in the growth plate morphogenesis. But clearly, what is particularly interesting from a therapeutic point of view is actually whether or not VEGF can inhibit is a tumor angiogenesis factor. This was really the very early one of the very early hypotheses. And actually, this is an experiment that was published exactly 20 years ago from our group. And actually, I'm showing you this very old data because essentially it was very important to initiate some therapeutic programs and what we did in this early study, we developed a monoclonal antibody using human VEGF, actually human VEGF 165 as an antigen. At the time, we knew absolutely nothing about the function of VEGF. The study was actually the genetic study was published 3 years later. So this was the very first in vivo study to probe the biology of VEGF. And so what we did, we implanted by a very conventional approach some human tumor cell line in nude mice, in immunodeficient mice, actually. This was a rhabdomyosarcoma, this is a glioblastoma, and this is a leiomyosarcoma. And then we treat the mice systemic with this antibody, this anti-VEGF antibody. At the time, it was a really astonishment, blocking of VEGF-A alone, seems to be sufficient to inhibit tumor growth. Like, for example, this tumor which proliferated very rapidly, which is the rhabdomyosarcoma, there was almost over 90% inhibition. But there was a clear spectrum of inhibition of tumor growth. And actually, this experiment was then verified in different animal models. This is, for example, is a model of liver metastasis of human colorectal cancer cells. The cells are injected in the spleen, and then they are transported by the portal circulation in the liver. And then you can see the liver of the animals treated either with the control antibody or with anti-VEGF antibody. And this is a parallel experiment done in injecting the tumor cells subcutaneous. So there was a very clearly an inhibition. And also this many other laboratories were able to obtain similar data, not only using antibody to VEGF, but either other inhibitor, for example, antibody to VEGF receptor 2, and very importantly by kinase inhibitor, which I will show you became important therapeutic as well. And also, this is another experiment which I think it's very simple, but makes a very important point about what really what you can achieve by blocking tumor angiogenesis. This is in this experiment, some ovarian carcinoma cells were implanted, in immunodeficient mice. Once again, the anti-VEGF antibody inhibits tumor growth. What was different in this experiment, that at this point, after 30 days of from the inoculation, the treatment was reversed. The control group received an anti-VEGF antibody and vice versa. So what happened is probably not particularly surprising. Essentially, the tumor started growing back, because by blocking angiogenesis, we didn't kill the tumor cells. We simply remove this break. And as soon as you withhold this treatment, the tumors grows back. So it makes a very important point in therapy about the need for a long-term inhibition of angiogenesis. Okay, so and actually this is another really very critical slide. This is one of the most important actually advances in molecular biology. Actually, essentially this illustrates the fact that there is a series of genes which are regulated by oxygen tension. Hypoxia results in upregulation of a number of genes. Actually, the first one to be characterized as such was erythropoietin. And then VEGF became very clearly was also upregulated by hypoxia. I think physiologically probably makes a lot of sense, because these genes are part of the same homeostatic cascade. If there is hypoxemia, the body responds by increasing erythropoietin, which in turn increases the oxygen carrying capacity of the blood, which and then a molecule like VEGF, which makes new blood vessels, enable the tissue to utilize this increased oxygen. And actually, this is essentially, without going into these details, essentially, this is, once again, it was a very important, a series of important discoveries. It showed there is a transcription factor, which is called hypoxia inducible factor, which binds to the promoter region of VEGF and other genes, in a very specific, responsive element, And this gene is tightly regulated by another genes which called VHL, with the von Hippel-Lindau. Those who study medicine probably are very familiar with this disease, the von Hippel-Lindau syndrome, which was until maybe when I was a medical student, it was this very mysterious, this very enigmatic disease where you have a combination of hemangioma, hemangioblastoma in the retina, in the cerebellum, as well as renal cell carcinomas. So there was this explosion of blood vessels. Now I guess we have a better idea why this happened, because normally the VHL inactivate HIF by a process of ubiquitination. And so if there is an inactivating mutation in the VHL, such as happens either in this genetic syndrome or in a number of tumors, there is this upregulation of VEGF even in normoxic conditions. So this is really, so once again, this is very important concept. VHL and HIF, they just regulate a series of genes. And given this background, actually, you probably be in a better position to appreciate actually these slides. This is show the expression of the VEGF messenger RNA in a number of human tissue using Affymetrix data. In green is the normal tissue, and then in red is invasive cancer. You can see there is expression in many tissues, which is greater in pathological condition. But there is a very clearly a signal which clearly stands out, which is in the kidney. It is essentially reflecting the fact that about 50 to 60% of renal cell carcinoma have this inactivating VHL mutation, which results in this very strong upregulation of VEGF, which provides a very nice molecular explanation. And so this cartoon illustrates some events which happens within the tumor microenvironment. In response to hypoxia, VEGF is released by tumor cells. And then it can activate, can interact with the VEGF receptor. Once again, especially VEGF receptor 2. But actually, it's very clear that it's not only hypoxia which regulates VEGF. There is a number of chemokines or cytokines, for example, like PDGF, IGF-1, and others, which can also stimulate VEGF. And also a number of oncogenes, like for example, an activating RAS, also result in upregulation of VEGF. So there is a series of events within the tumor microenvironment which result in VEGF upregulation. And so maybe in the mid-90s or so, we thought of developing, a therapeutic based on this work on VEGF, and actually in 1997 we initiated some clinical trial. But before showing a clinical trial result, I would like to show you actually what was known about this angiogenesis inhibitor. Actually, There have been already many previous attempts, and the picture was very, very disappointing, very discouraging, actually. All of this approach had been unsuccessful in the clinic. For example, you may have heard about MMP inhibitor, matrix and metalloproteinase inhibitor, which seems to be inhibit some very fundamental mechanism of invasion of angiogenesis. To the extent in the 90s, virtually any pharmaceutical company had the program in MMP inhibitor. There was this expectation that this would be very successful therapeutics. Unfortunately, that was totally disproved by the clinical data. All of them, fail in the clinic. Actually, they were detrimental to patients. They reduce, survival for reasons which are not yet, completely clear. And then there is another series of molecules like endostatin, which at the time became a very famous molecule based on an article in the New York Times, which quote, Jim Watson as stating that a molecule like endostatin will cure cancer in 2 years. Actually, of course, it did not happen. And then there was a series of other molecules, which once again, and so once again, it was a really very very tough, kind of very difficult to convince anybody that angiogenesis could have a beneficial effect. But once again, what we did, we humanized this monoclonal antibody, which I described you previously. Humanization, I don't know to what extent you're familiar with this process, but essentially it's a technology which is used, to reduce or overcome the immunogenicity of murine antibody, because this was the original antibody was a murine monoclonal. You don't want to give a murine monoclonal to humans because it's going to be quickly immunogenic. So this is by the genetic engineering technology which allow you to introduce the regions which are important for binding antigen into a human framework. And the final product is about 5% murine and 95% human. And so this is what we did, and this antibody became known as bevacizumab or Avastin. And once again, began a clinical trial in 1997. And actually, I will show you actually the first definitive study, which actually was published in 2004 in the New England Journal of Medicine. Actually, was presented at ASCO, this American Society of Clinical Oncology meeting, actually 10 years ago, actually in June 2003. And actually, everybody expected that this would fail, just like any other angiogenesis in it. But instead, it was very, very gratifying to see that there was actually survival benefit. This was patients with metastatic colorectal cancer, previously untreated metastatic colorectal cancer. This is a lethal disease, unfortunately. It's an incurable disease. But this treatment was able to increase, patient survival by about 5 months, which at that time was the longest survival benefit conferred by biological agent. Obviously, this is not a cure for cancer, but result in a benefit. And now actually bevacizumab was approved, actually. It showed benefit, even in multiple line of therapy in colon cancer. Actually, I'm not going to go over in the details. But even patients which in relapse colorectal cancer, which been treated in first line and then relapse, they still show a benefit, with this therapy, which now still is the main standard of therapy. I want just to show you actually one very recent piece of data. I will add, that this treatment is shown, but has been approved also in lung, cancer, in renal cell carcinoma, in glioblastoma. Actually, the result in breast cancer was mixed, but this is actually very recent data in cervical cancer, which is a tumor which is highly aggressive. Actually, even early study showed that the appearance of angiogenesis, this is in a colposcopy, is a kind of hallmark of invasiveness, and the study was very recently presented. Actually was conducted by the NIH by this. The study is called GOG240. And once again, it shows a survival benefit by adding bevacizumab, to this therapy. And once again, I'm not going to go into it. This is another example, actually, of what I was telling you at the very beginning, that the duration of the therapy is very important. In the study, comparing actually a short-term treatment with the bevacizumab, this is patients with ovarian cancer, versus a long-term maintenance. And once again, the greatest benefit is very clearly seen when the bevacizumab treatment is done, for a prolonged period of time, because for obvious reasons. And once again, this is I'm not trying to suggest, that, block, this signaling pathway with an antibody is the only way to do so. There is, I was alluding at the very beginning, there is a multiple way to inhibit, the system. You have not only an antibody to VEGF-A, but you have, antibody to VEGF receptor 2. You have also a soluble receptor. One has been approved in the United States. It's called VEGF, trap, which is a soluble receptor. And also there is a series of kinase inhibitor, which inhibit VEGF receptor signaling, actually. This molecule are not totally specific because it would be very difficult to have a high level of specificity. They almost invariably they also block, related receptor, like, PDGF receptor, KIT, and probably many others. And so there is and as of today, there are as many as 10 different, VEGF, pathway inhibitor, which has been approved, for cancer and for a series of diseases. I'll show you toward the end of my talk, actually. And actually, this is illustrating some of really the most, the most, salient, at least the most, extensively investigated, signaling pathway which result to angiogenesis, which are therapeutic targets. Not only we have the 3 VEGF receptors which I described you, but it's also another system which called TY2, which is another tyrosine kinase receptor, which is the ligands of the angiopoietins, angiopoietin 1 and 2, and this is also there is some attempt to block this molecule therapeutically, and also we have some integrin antagonists, like, because integrin are essential component of the extracellular matrix, and there has been various attempts to inhibit several integrins, even though as of today they not be very successful. Actually, this actually is an interesting example. Actually, a very good idea to now to be very complicated. This is work done by my former colleague when I was at Genentech. They identified a molecule in the a gene called EGFL7, EGF-like 7, which it's a highly endothelial cell specific molecule. Some early study in the zebrafish, showed that blocking this molecule had a dramatic effect, in vascular development, and that led to the idea this could be a very important therapeutic target. And once again, this illustrates how biology can be very complicated. What happened is that, in a few years ago, there was this explosion of microRNAs, which I'm sure are very familiar. There is in particular one is called miR-126, which is now seems to be very important in vascular development. And it is miR-126, almost diabolically, is encoded by the same locus as EGFL7. So this early study, which actually which we did a knockout of EGFL7, which resulted in this dramatic phenotype, unknowingly and inadvertently also inactivated this microRNA. And then it became known actually that this microRNA is the key player in this system. So once again, there was really, and there is some evidence actually that all the phenotype which was initially attributed to this molecule actually was due to the microRNA. So once again, illustrating the complexity of biology. And that actually, the slides illustrate a series of actually the molecule I was alluding to of this, which either been approved by the FDA or are in different stage of clinical development. I don't expect anybody to read all these things because there are too many. But besides, bevacizumab, which was approved, in 2004, there is a series of kinase inhibitors, like you're probably familiar with the sunitinib, sorafenib, axitinib, and many others. And now there is a series of other molecules, which most of them target, the VEGF, pathway, but not all of them, And so time will tell how well they will do. So one point which really it's worth mentioning, this is an area of molecular therapy, personalized medicine. Actually, the goal, everybody strives to create a therapy which target only the patients which are most responsive to treatment, and not treating the others. And so most of the target therapy work on a very narrow patient population. For example, the best example is Gleevec, which target patients with CML, or Herceptin, which target patients with advanced breast cancer with HER2 upregulation. In the case of a VEGF inhibitor, it's been, so you have a very precise biomarker which allow you to select patients. Until now, it's been very difficult actually to identify a biomarker for VEGF inhibitor. But what is very interesting that it's been possible to demonstrate some beneficial effect even in this completely unselected population, treating all comers. Once again, usually your biomarker is the target itself, like VEGF. To not be the VEGF is a prognostic value. The higher the level of VEGF, the worse is the prognosis. It's been very difficult to show that the VEGF is representing actually a biomarker. And so there is probably a number of complexity why it's difficult to identify the biomarker for a molecule which inhibits angiogenesis. It's probably conceptually much easier for a molecule which inhibits some tumor cell autonomous phenomena, like for example a mutated oncogene, because there is a lot of complexity of your target cell type, which such that it's very difficult. Until now, it's been very difficult to identify this biomarker. There is actually one very recent data actually from my group. Actually, we've been able to identify a gene signature which is seems to be predicting the response to VEGF inhibitor. Actually, once again, the paper was published very recently. But to make a long story short, is that we identify a set of 26 genes, which seems to be interestingly in the mostly gene targets of VEGF. They are molecules which are regulated by the VEGF receptor. And essentially, patients, this is from a colon cancer trial in bevacizumab. This is a purely retrospective analysis, so that it will need to be validated, in a prospective study. But once again, patients which are very high level of this, signature, they show a greater responsiveness to bevacizumab. But once again, this will need to be validated. So the slide makes another point. Actually, this is really that all cancer therapy, without any exception, virtually without an exception, result in resistance. This is actually one of the wonders of molecular medicine, which is a drug which is called Zelboraf, which is inhibits BRAF, which is in a subset of patients with the melanoma. It inhibits this mutant RAF. You can see this before therapy. This is a patient which has lots of lesions, and this is a patient which after treatment with this inhibitor seems to be completely clear. And then several weeks later, actually, this is which almost inevitably happens. There is a relapse, there is a recurrence, because there is a mutation in the same pathway. And there is actually another really example I would like to make. I don't know if this is another really very intriguing example of therapy, of targeted therapy, which is there is a subset of patients with the non-small cell lung carcinoma, which have actually a chromosomal translocation, which result in activation of a kinase called alkynokinase, something very similar to what happened in the CML, in chronic myelogenous leukemia. There is a BCR-ABL, which result in this kinase. And this is something very similar. Even though in CML and very intriguingly, actually, this alkynopositive lung cancer can be treated with a specific inhibitor of this alkynokinase, which is called crizotinib. Now, once again, even in this case, crizotinib, which is a very dramatic effect at the very beginning. There is a very clearly strong response even in phase 1. But overall benefit perhaps is not as great as you believe. There is about 4 to 5 months increase in progression-free survival. There is actually, which is a very respectable benefit, but once again, is far from that, cure for cancer that everybody is dreaming. So once again, so show that, really how much more work needs to be done, in this field, to advance, patient care. So once again, it's not surprising that even the VEGF inhibitor have, this problem. There is a resistance, and actually there is actually reduced responsiveness. I don't know what we are doing with time, actually. So this is clinical data. We show there is a benefit in patients, but certainly we're not curing cancer. Some patients actually relapse. certainly suggested it would be a good idea to go back to, the laboratory, try to understand what could be the mechanism resistant to angiogenesis inhibitor. And actually, there is this slide illustrated the tumor microenvironment, which multiple prayers, multiple cell types, and there is a very large number of possibilities. You can have, for example, different angiogenesis factor being produced by tumor cells, which of course could not be blocked, by VEGF inhibitor. But what we've been particularly interested in the idea that the tumor microenvironment, and in particular bone marrow-derived cells, they can play a role in this resistance. Actually, this has been, my lab is focused on this idea now for a number of years. Actually, we need to create, some models to study, actually, all of that, So what we did, first of all, we could not use an antibody like a bevacizumab because that is very species-specific, blocks only human VEGF. So you can study in human xenograft, in immunodeficient mice. Instead, we want to study in mice with an intact immune system, And so we developed some new antibodies, some which cross-react by phage, technology, by this antibody cross-react, inhibit equally well mouse and human VEGF. And so actually we identified like a spectrum of cell line, which actually these two in red, TIB6 and B16F1, we call them the sensitive model, which are inhibited by 80 or 90% by VEGF inhibitor. At the end of the spectrum, we have, this tumor types like the Lewis lung carcinoma and the EL4, which show certainly reduced responsiveness to VEGF antibodies. And we're hoping that by comparing and contrasting this cell type, we could actually learn something. And actually, the story, we published a series of papers in that, so I'm not going to burden you with lots of data, actually. But what we found, we were able to identify this kind of signaling pathway. We found that there's some myeloid cells which are called CD11B GR1 myeloid cells, which actually represent a broad spectrum of cells which include neutrophils, macrophage, and dendritic cells, as well as some cells which are actually called myeloid-derived suppressor cells. Actually, an investigator from Padova, now in Verona, Enzo Bronte, has been a kind of the leader in this field. Actually, these myeloid-derived suppressor cells are at least a subset of this CD11B GR1 myeloid cells, which is in human. The precise counterpart in human is less, precisely established, but probably there are immature neutrophils, which actually do that, And so we discovered actually the cells, play a very active role, in this resistance to VEGF inhibitor. We discovered that actually one major myeloid, growth factor, which is called GCSF, or granulocyte, colony stimulating factor, essentially act on the cells and not only mobilize them, is a very potent, powerful stimulus to mobilize cells. Also induce some very specific pathway. There is in particular one pathway, actually our lab had previously played a role in identifying. It's a small protein called BV8, which was initially identified as a frog-derived protein, actually by an Italian group, by Lucia Negri in Rome in collaboration with another group. But we characterize this molecule as an angiogenic factor as well as an hematopoietic factor. And GCSF is extremely powerfully induced of this molecule by a statin 3 mechanism. And then BV8 actually induces angiogenesis and myeloid cells mobilization in a manner which is independent from VEGF. So that's what we found in this study. And actually, this is what happens if you block, BV8, and actually in a colorectal cancer model. This is a micro-CT angiography which gives you an idea of, the tumor vasculature, And once again, an anti-VEGF antibody is, perhaps not surprised, is the strongest effect. The anti-BV8 antibody also has some significant inhibitory activity. And actually, what is very intriguing, actually, this is perhaps is not only limited to mice. This is a section of, of a colon carcinoma, which you can see there is a lots of, tumor-infiltrating neutrophils, which is strongly expressed, this protein actually, and I will show you some later development of the story actually of the neutrophils which can play a role in colon cancer. And so, given that GCSF seems to be our driver, the primary driver of this phenomenon, actually, we're interested in this molecule. GCSF is a fundamental regulator of granulocytopoiesis, which of course is a very essential phenomenon. Also, it's known as a very important therapeutic. It's called Neupogen. I don't know what it's called in Europe, but probably very similar name. It's a major product. Is used to treat patients with a chemotherapy-induced neutropenia, which otherwise cancer patients would succumb to chemotherapy. And what is perhaps less appreciated is the fact that GCSF itself, is a tumor-derived molecule which have a pathophysiological role. It's known to be produced by a variety of tumors, mostly by bladder cancer, breast cancer, and others. Give rise to a paraneoplastic syndrome, which actually is characterized by an extremely high level of neutrophils, to the extent that they're called leukemoid, reaction. In previous, it's been shown that this, patient with this kind of high level of GCSF have an extremely poor prognosis. And so we're interested, in establishing why GCSF is produced by tumor. Actually, a series of studies. The study was published very recently. Actually, we identified some tumor model which have a very low and very high production of GCSF. And we're able to show that actually RAS, the RAS signaling pathway plays a very critical role into that, resulting in activation with transcription factor, which is called ETS2, which in turn, upregulate, GCSF, and actually, if you introduce, actually, I think the symbol is a little bit, corrupt, but if you introduce, for example, a mutant RAS, in a cell line which does not produce a GCSF, that results in a very dramatic increase in GCSF, production. And actually, there is a therapeutic implication in all of that. Now there is inhibitor of, actually, once again, this is MEK, this is the pathway. There is a multiple kind of players, There is a RAS, MEK, and ERK. There is an enormous level of complexity, but there is a general agreement on MEK, can be a rate, limiting step, And so we had a MEK inhibitor, which right now is in clinical trial, which is called, GDC 0973. And it was very, very nice, very gratifying to be able to demonstrate that actually this MEK inhibitor reduce, GCSF level in a dose-dependent fashion. This is one of those cell lines. Actually, we're able to, in this paper we showed that about, almost 50% of a human tumor cell line, which with activating RAS pathway, producing GCSF, which is inhibited by this MEK inhibitor. And so actually, actually, we're able to show actually in this experiment, actually, this is a pancreatic cancer cell line, which of course there is a mutated RAS, And actually, the combination of a MEK inhibitor with an anti-VEGF result in a greater potentiation of this anti-tumor activity. And actually, in the course of the study, actually, we stumbled in another very important area of biology. Not only what I've shown you so far, the myeloid cells are part of the innate immune system, naturally. But there's probably a very important component of the immune system, which is include also adaptive immune system, which is the IL-17, which is produced by Th17 cells, which is a subset of the T cells, which has been implicated, in inflammation, in autoimmunity, in response to bacteria, and actually they are very critical for defense, to bacteria. And actually, this was recent work done by a postdoc. This work actually was published, it's been published in this issue of Nature Medicine, in the September issue of Nature Medicine. We're able we look at our original model of sensitive and resistant cell line for what kind of cytokine they produce. It is one which is very clearly stands out. That is actually IL-17. For example, if you compare the EL4 tumor with TIB6 tumors, there is actually a lot more IL-17 produced by the first. Actually, there is a lot of that. And so we follow up, this biology, and actually we're able to show that actually IL-17 signal seems to be very important actually for myeloid cell recruitment. Actually, we took advantage of some IL-17 receptor knockout mice, so we could completely inhibit the signaling system. Immunologists have known for a long time that Th17 cells result in attraction with neutrophils, And I think that's probably the explanation is that, the IL-17 is stimulating stromal cells to produce myeloid growth factor, like GCSF. And actually, we're able to show that actually this function of IL-17 is also, is strongly additive to VEGF inhibitor. Actually, this is implanting some of these resistant tumors in IL-17 receptor knockout mice. And once again, this is the effect of IL-17 alone. There's a very little effect. But combining anti-VEGF with in this case, anti-VEGF has a much more dramatic effect. Something very similar can be obtained with antibodies, even though perhaps less effective than with the genetic inactivation. And actually, this is another example. This is an orthotopic model lung carcinoma, which tumor cells were orthotopic injected into the lung. And once again, in this case, there is a very clear additivity between anti-VEGF, alone, which has some effect, and as well as anti-IL-17. But the combination results in a much more profound inhibition. And actually, what has been very interesting is that being able to look at in this case a human section, a human tumor section, there is a very strong correlation between the presence of the Th17 cells, and BV8 expression in neutrophils. I'll show you at the previous that this colorectal cancer cells have tumor infiltrating neutrophils with producing BV8. And now it's very very intriguing to see also there is strong correlation with the Th17 cells, because it really fits very nicely with our hypothesis. These cells that result in attracting neutrophils. And it has been known for a long time that Th17 cells are a negative prognostic factor in colon cancer. And actually, this will summarize some of the studies. Actually, once again, the Th17, not only is produced by certain tumors, by some certain leukemias, and that EL4 is one example. But more physiologically relevant is that Th17 is a product. IL-17 is a product of the Th17 cells. So in this case, there is the very clear crosstalk between the native immune adaptive system, which results once again in increased angiogenesis. I don't want to go over time, but I think it would be a big omission on my part if I didn't show you at least last 5 minutes, which has been certainly the most successful application of anti-angiogenesis, even more than in tumors actually. I think many probably are very familiar with the fact that there is a number of diseases which result in blindness, like diabetic retinopathy or age-related macular degeneration. But there is a lot of blood vessels. Actually, this is one of the scheme. This is the fundus of patients with a proliferative diabetic retinopathy. You can see lots of vessels which kind of irradiate from the around the head of the optic nerve. The reason why these vessels are so destructive, so dangerous, because they are very fragile. So they have a propensity to bleed and to leak, and so that can destroy the photoreceptor. In the extreme case, it can result in retinal detachment and blindness. And actually, this is quoting one of the really these giants of the past, which is Michelson, who was already in 1948. Basis solely on kind of a clinical observation, is speculating that the retina produce a diffusible factor, which was called factor X, perhaps not very original, factor X, and there has been lots of search for this factor X over the last decade, and actually there was also very clinical data which is clearly implicate ischemia and hypoxia in the production of factor X, given the fact, actually. And actually, this raised the question. Given the fact, in the early 90s, mid 90s, it became clear that VEGF is regulated, by hypoxia. Is VEGF in any way related, to factor X? And actually, the study was published now, 1994. Actually, it was a collaboration between my group, and a group at the Harvard, the Joslin Diabetes Center, which had a very extensive collection of, eye fluid sample for patients with a different disease actually. And we were able to show that there was an extremely tight correlation between the presence of VEGF in these eye fluids, like in aqueous and vitreous, and proliferative disease. Like this is the proliferative diabetic retinopathy. This is very high levels, And what is very intriguing when the disease becomes quiescent, which is part of its clinical course, VEGF is almost undetectable. And VEGF also elevated in other diseases like, retinal vein occlusion, etc. But this was only correlation. What was, unfortunately, the slide has been cut. But the point is that you need to have an animal model, to have, to test, this hypothesis. And this was a primate model of, ischemic retinal disease of iris and neovascularization. And you can say this is the control. This is a control eye, which this intense, neovascularization in the iris. And this is said that the eye of an animal treated with the intravitreal with the anti-VEGF antibody resulted almost this seems to be a very complete inhibition of angiogenesis. And actually this led to the idea that this treatment could have a beneficial effect even in eye disease. But there is a disease which is eye intraocular neovascular disease, which was even more of an unmet medical need than ischemic retinal disease. And that is age-related macular degeneration, which is actually one of an extremely complex disease. Actually, it's the leading cause of blindness in the elderly population. Actually, and now it's recognized there is like a spectrum of disease, like some early stage, and AMD, late stage advanced AMD, which can lead to neovascularization. Now there is a very elegant genetic studies actually we implicate complement mutation in the complement, especially complement factor H. There is a defective inactivation in the complement which results in inflammation. But the point is that neovascular AMD accounts for about 20% of the case of AMD, but is responsible for about 80% of the case of blindness. And the reason is once again is very way too familiar. You have growth of blood vessels come from the choroid in this case, go through this structure which is called the Bruchino membrane. And these vessels have this known sequelae of events. They are very fragile, so they bleed, they leak. In this case, even a very discrete lesion, like in this case in the macula, can be very destructive because it can destroy the photoreceptor which are necessary for visual acuity. Actually, this is the way a patient with the AMD is supposed to have relatively sparing peripheral vision, but the central vision is very severely affected, because actually these patients could be otherwise healthy. They're unable to write, they're unable to read, to drive it. So it's a devastating disease because they have very poor quality of life. And actually, there were very few therapies which have been available for this treatment, actually. And actually, in the late 90s, actually, even though this at the time was an extremely controversial idea to propose, we thought of developing a VEGF antagonist even for age-related macular degeneration. Like I said, at the time, the case for a VEGF for AMD was much less clear for the ischemic retinal disease. It was actually believed that there is so many mediators of inflammation that it would be really hopeless that blocking only one factor could have a beneficial effect. But once again, sometimes biology surprises you. This is actually phase 3 data in patients with the aggressive form of AMD, which is called classical form. There is this florid neovascularization. And patients were treated in this trial with the monthly intravitreal administration of, actually it's a VEGF inhibitor, actually it's an antibody fragment, which is an FAB, which was injected monthly. And then patients were followed by measuring visual acuity. So the green line actually is not a placebo. Actually, this was at that time a standard therapy, which is the photodynamic therapy. You can see there is a very transient increase in visual acuity, and then there is this seems to be a complete visual loss, which is going to be very very similar to a placebo. Instead, actually, to everybody's surprise, both arms of ranibizumab, this is the name of this antibody, called also Lucentis, show not only there is slow down visual loss, but actually about 40% of patients there is increase in visual acuity, which seems to be maintained for over actually 2 years, which is the time of the trial. And actually, this illustrates some of the concept of vascular biology. Actually, we've been able to which perhaps can explain this benefit. One thing which is really very unexpected, actually, a little bit contrasted with the tumors. Blocking VEGF, in this setting, result in a virtually 100% inhibition of angiogenesis in a very large number of patients, which is actually in some there is even a regression of blood vessels, which is of course very different from tumors. Perhaps the fact that tumors have so many mechanisms of genomic instability, they are able to activate pathway, evasive and adaptive pathways, which evidently this pathology, a non-malignant condition, is unable to do so. So there is this very strong dependence on VEGF. And actually we can say it's very gratifying to be able to say that the impact of VEGF inhibitor has been very dramatic. Actually, there is some recent study which actually measure the outcome of patients before VEGF inhibitor were available with those who had been optimally treated with this agent. In this case, AMD would no longer be the leading cause of blindness in the elderly population. And actually, and I think this is certainly has been one of the most dramatic success of anti-angiogenic therapy, which has certainly changed the course of this disease. Not only with the ranibizumab, but now there is a 3-VEGF inhibitor which is widely used. And one is bevacizumab, which is widely used off-label. It's not been approved, to treat this, but it costs much less than bevacizumab. It costs maybe 1/20th of the cost. So it's still very effective. So it's been certainly. It was unintended, of course, but this is really certainly has added benefit. It is another drug which is a aflibercept, which is a soluble receptor, which also inhibits VEGF. It's essentially the same efficacy as ranibizumab, but so there is at least 3 therapeutic options. And certainly there has been a benefit in multiple tumor type, which and now the VEGF inhibitor represents standard of therapy. But certainly. There is a lot of work that needs to be done for, especially in tumors, because we don't really understand, we don't know the biomarker which allow to select, patients such as this therapy would have even greater impact, And also, still, there is a lot of interesting combinatorial therapy. I was alluding in my talk to some of those, like, for example, immune therapy. Now, there is evidence that, some powerful immunotherapy, not only IL-17, but even like a PD-L1 or CTLA-4, could have an additive effect with the VEGF inhibitor. And there is a lot of fundamental research which needs to be done, actually, to understand what could be some pathway which result, in angiogenesis. And finally, I would like to thank many colleagues and collaborators which have given really very essential contribution to this work. So I'll be very happy if there is time to answer any questions.
Thank you very much for this beautiful presentation, which is now open to discussion. Shall we have discussion in English or
either
take questions?
Either way, that's fine. Benissimo, benissimo.
Allora, in italiano e in inglese. Maybe I'll start off with a couple of questions. The first one, the VEGF receptor, is it exclusively expressed in the endothelial precursors, or do you have also in some expression in other cells?
A very, very good question, actually. Actually, VEGF receptor 2, to the best of my knowledge, is still largely expressed, actually they're expressed in hematopoietic, progenitor as well, because once again, hematopoiesis and angiogenesis are very tightly linked, actually. It's very cool. There is actually an Italian group, Cesare Pescle or Pescle, in Padova, studying KDR, VEGF receptor 2 in hematopoietic progenitor, in hematopoietic stem cells. But certainly VEGF receptor 2 is very clearly expressed in hematopoietic and endothelial lineage. VEGF receptor 1 instead is known to be expressed also in macrophage, in monocytes, and more recently there is some evidence it can be expressed in tumor cells, in some tumor cells, where it results in a sort of, cell autonomous growth, in this case it behave more like a typical oncogene, like the EGFR, etc. But it's very unusual. Maybe only a subset of tumor cells have that. So I think it's still, the system is mostly, we learned that there is nothing exclusive in biology, but it's largely selective for endothelial cells.
So you can imagine that it really drives only vascularization and not utilization. I'm fond of mitochondria, so I was trying to find out whether the same signal that induces vessel formation can induce a pro-oxygen usage phenotype in the target. But that's
I don't believe that there is lots of evidence for that.
You mentioned that the renal cancer, colorectal cancer, glioblastomas, and so on. You know that the multiple myeloma is an hematological malignancy where VEGF plays a central role. Is there any possibility, can you comment on the possibility to use VEGF inhibitors in this disorder?
Well, I think it's a good idea. Of course, there is in a number of models of myeloma. I know there has been several clinical trials done. I don't believe any was sponsored by Roche or by Genentech. There was one done by the Mayo Clinic, etc. There was some early promise, but I think they're competing with a therapy which is already very effective for multiple, well, at least it's...
You know that lenalidomide, which is an immunomodulatory drug which interferes with VEGF, is effective, but I am not aware of other experience with VEGF inhibitors.
Once again, I think there has been some trials, some relatively small trials, and I really cannot tell you exactly the outcome, but there has been attempt. But once again, the drug that you mentioned, which has very broad spectrum, inhibits angiogenesis, inhibits TNF, inhibits almost everything, they're believed to be very effective. So that's maybe what is really, perhaps, reduced the enthusiasm of testing this drug, and focus on instead with disease where there is more an unmet medical need. But I think it's a very, very, very good hypothesis, etc. So that I'm not aware of any data.
Marco?
I have a question that is slightly related to what has been asked before. So we work on cardiac neurons and sympathetic neurons, and VEGF also is a trophic factor, which is both released to the neuron, is a trophic factor in the neuron, and apparently released by the neuron. I was wondering whether therapies with VEGF antagonists have, or do you have an idea of whether there is cardiac dysautonomia in a way?
I think you made some very important point, because actually I didn't mention in the interest of time. There is actually, probably for the audience, very intriguing data which suggests that neurogenesis and angiogenesis share a number of molecular mechanisms, like for example the ephrins, the neuropilins, etc. The molecule which was initially characterized as axon guidance receptor, and now they bind VEGF. But in terms of this effect of VEGF, I think in the nervous system, I think the data are not so clear to me, because there is lots of data in vitro, but the in vivo data, the genetic evidence perhaps does not suggest it's a major effect. There is I'm sure you're familiar with the elegant study by Elie Keshet, which reported VEGF is implicated in being intelligent, in memory, etc., etc. I tend to be slightly skeptic about this data, because once again, between the mouse and the human, there is a lots of difference, and it's extremely difficult to dissect effect on endothelial cells versus effect on neurons, because endothelial cells produce, as you know, a number of neurotrophic factors like BDNF and many others, which can affect the neuronal cells. So it's very hard, to kind of, to dissect, these two events. But you're absolutely right. There is evidence, that VEGF in vitro can have effect on a variety of, the neural cell type. Whether this is translated to a broad in vivo effect is less easy to say. Naturally, this is the data by Peter Carmelitin, which you're probably very familiar with, that implicating VEGF in ALS, but I don't believe that the data have really borne out.
Before we carry on the discussion, I would like to encourage the students to ask questions, because the audience is physicists, chemists, mathematicians, so I think they may be intimidated in asking a question, feeling a little bit outside of this. Well, I think it would be great if a question which necessarily cannot go to the molecular detail, but maybe goes to conceptual issues, will be asked by students. So please don't be intimidated by all these medics asking the questions. If you have any curiosity, ask the question, and not another medic.
I'm the younger fellow. You start as endocrinologist.
Yes.
So I wonder if...
Everyone has a defect, come on. Everybody has defect.
Are you an endocrinologist?
Yes.
I see.
So, do you have any information about specific receptor in endocrine cells of tumors of VGF? There was some idea in the past about it. Some of the receptor would localize, especially.
Well, you know, I'm not really aware of any specific receptors, you know, in endocrine cells. But certainly, endocrine glands are highly vascularized. You know, there is, of course, like a thyroid and other tumor, which is extremely high. In this case, I think, you know, VEGF has been clearly implicated as one of the mediators, for example, in thyroiditis, you know, thyroid, you know, tumors, and probably maybe several others, you know. So I don't know if there is any specific, any special role, you know, in the system as opposed to others. But clearly, I think it's this was what initiated all this idea, actually, looking at the glands.
And you in the von Hippel-Lindau patient, the treatment with this inhibitors is worthwhile to check.
I think there's been some. Actually, you're right. I'm not aware of any large, you know, clinical trial. Also because it's an extremely rare, you know, disease. You know, the von Hippel-Lindau. I know that, you know, some patients have been treated, you know, with the VEGF inhibitor have certainly reduced, you know, the size of, you know, let's say the congestion, you know, the edema. They reduce the vascularity of, for example, hemangioma in the retina. So there is probably some small study in the literature, some small trial which documented that. But once again, the disease is so rare that it's very difficult to envision that a large, you know, company is going to study, you know, that, you know.
Do you think that vessels grow differently in different settings? Like, for instance, in epithelial tumors versus non-epithelial, which means, you mentioned briefly, the crosstalk between vessel growth and extracellular matrix.
Yeah. Well, there's of course, a very fundamental question, actually. The fact that tumors grow differently in different tumors. There was actually a seminal paper published in 1927, actually, already. The first, you know, to illustrate, you know, that, you know, that actually, to make, you know, this idea, was Lewis, actually, suggested that the tumor environment, they didn't call it microenvironment, they just called the environment is a critical role in determining because it was very clear the vascular pattern is different in different tumor types. But still, whatever the factor, which still is not totally clear what it is. There is a lot more of a fundamental biology which we need to do, because there are some molecules, some essential players, like, you know, of course, like, you know, some growth factor, like, you know, VEGF and probably others. But there is still lots of other molecule which is very critical in determining this diversity, which I think we're only scratching the surface now. I'm not aware of anybody who come up with a very convincing hypothesis why, like, epithelial vessels in epithelial tumor are different from those in a different tumor type.
I like students' questions, so I'll pretend I know. One of the the dogmas that students learn is that tumor cells are anaerobic. I mean, that Ehrlich effect. So it is amazing that the anti-angiogenic factor is so remarkably successful, because in principle, in the growth of the tumor, you'd imagine that this is not exactly the right target. So, can you make it a broader explanation of what?
I think it's a very important point. That's maybe, you're right, maybe this is probably a reason why the effect in tumors, probably they're very much, you know, dependent on the tumor type. They're not really uniform, because maybe the tumor metabolism, you know, there is a metabolic adaptation such that tumors, you know, maybe even less, you know, dependent on angiogenesis. This is probably one of the and actually, I will add, you know, more. Actually, there is some the tumor seems to be so clever, you know, overcoming the physiological mechanism that, you know, in certain tumors, like for example in a subset of melanomas, uveal melanoma, the tumor cells can actually line, you know, blood, you know, filled cavities, essentially replace the endothelium. And so essentially they don't need anymore the conventional angiogenesis. There is some recent studies published recently claiming, at least showing that some tumor stem cells, like glioblastoma stem cells, can differentiate into endothelium, like working even by an Italian group, by Ruggero Di Maria and two other groups, so suggests the complexity of tumors and the difficulty, in this, in therapy, in targeting the endothelium therapeutic. That's maybe why a non-malignant, a series of non-malignant disease like intraocular neovascular disease has such much greater responsiveness.
I have a very non-medical question. Going back to the age-related maculopathy, will you succeed before the bionic eye guys, or will the bionic eye guys will succeed before your drugs?
I don't know. It's very difficult to tell. I think before we have this kind of, it will take probably a very long time. I think in the therapeutic effectiveness in macular degeneration is very, very, very clear. Actually, once again, this is in the neovascular age-related macular, in the neovascular stage, when you have all these blood vessels that grow, and this is the most deadly. But there is another stage of the disease which is called the dry form, with the geographic atrophy. We have actually death of the retina, because once again it is excessive inflammation. This would not be inhibited by this treatment. So there is a real room for other approaches. So there is a really many different avenues to achieve therapy. Maybe the bionic eye one day will do, but I don't know that. Maybe not in my lifetime.
So following this, your question, would you imagine that therapy in a few years should be advised even before proliferative retinopathy arrives in diabetes? Say, when you start to see some early changes of the retina, you'd suggest that...
Well, diabetes is very complex, because once again there is already an effective therapy, which is a laser. That's why this initial study with the VEGF inhibitor was done in macular degeneration and not in diabetic retinopathy, which is the more established condition, because there is already an effective therapy there, which is laser. But actually, the macular degeneration is another very interesting example, because about maybe close to 50% of patients, there is a mutation in the complement. There is a different mutation which is in the complement factor H, which has been shown to be clearly a risk factor for at least for the dry form. That in that case you may envision in the future if this drug, which there is a lot of drugs which target complement, which are in clinical trial. There is actually very recent and very early stage positive data. So one day if this therapy becomes established, then that could be the ground for initiating a therapy solely based on the genetics.